Hello, everyone. Welcome back to part 2 of our ENCODE lesson. Here, we're going to actually just do a quick survey of what's going on here with this image we just ended the background lecture on. So we mentioned before, we saw this picture, but now we're actually on the ENCODE portal. The link is in the description, but it's really is quite as simple as typing in your favorite browser, ENCODE portal, which will come up as the first. That's a bad test of this. There we go. Small little glitch, but we're back to the same spot we just were. So the link is in the description, but it's just ENCODE portal in Google, and you'll find it. Okay, so you can see there's all the different types of data. DNA methylation is methylome. Some of these words are a little confusing, but I like this diagram they have down here, which is also interactive, where we talked about promoters, where DNA binding proteins will come together and help turn on genes or help turn them off, and work together to regulate gene expression in a cell. But there's also enhancers. We'll talk about those more. But ENCODE has basically laid this out like the genome would think, that there's a promoter region where is like the on-off switch for a gene. And then there's the gene itself with these transcripts with RNA binding, and transcriptome just means the RNA output of this promoter or anywhere in the genome. RNA can come from enhancers as well, and we'll get into that later. If you want to see the introduction lecture, we talked about that a little bit in the last class. But really all of this you can see are clickable, and it'll tell you how much data is available, what news there is, etc. But what we can also see here is assay categories. So that's another way of saying these same things, where chromatin interactions means DNA binding. So if we just click here, we can see there's 10,198 DNA binding experiments of where things are bound on DNA. So for this class, that's where we want to go. You can either click there. We'll go to chromatin interactions here, and then click on DNA binding within that. And we're transported to this new page here, which I'll make a little bigger so we can see this better. And here on the left is a bunch of information that is really important. This is often not recorded in most people's experiments, but ENCODE has a way for you to toggle through the things you're interested in, and it'll produce a data set for you. And that's exactly what we're going to do in this class. Is we're going to look at the DNA binding. We've already picked that, shown here. This is sort of that initial menu, but we picked DNA binding to get into this area. And so in this class, we'll just toggle the things we'll do, and we have the R markdown file in the link below, and we'll get into that in the third part of the class. But we'll click, we want the TF or DNA binding Chip-seq. We want histone Chip-seq, which tells us how the DNA is packaged into these on and off regions. And then we want the controls for that Chip-seq. But we're only interested in one cell type here. So well, first of all, we're going to stick to human. So we'll click that there. So now we've it's like ordering on an online menu, if you will. So we've picked human, and there's a bunch of different cell lines, and we'll talk about this later. Last year we did K562, and this year we're going to do HepG2. K562 is a blood cell line, and HepG2 is a liver-derived cell line. And so we can ask a lot of the same questions we did in the last class and see if they're different in different cell types or how these properties change. So we're just going to click HepG2 because we're going to do this in one cell type. And look, we get this nice thing. We got 651 Chip-seq things, which is actually less than it sounds because there's also replicates and other controls, which we'll talk about later. So we picked HepG2. We're going to also pick different read lengths. And here is where we would first go to our class, our markdown file in the, in the class 2021 repository, we can see this O1 encode R markdown file. And here I've already outlined some of the information we're going to pick. So we're going to pick read lengths of 100, 76, 75, and 36. So let's start from smallest to biggest here. We'll pick 36. Then we'll pick 75. Oops, sliding down here. Then we'll scroll up to 75. 76, which should be close by. 76. And 100. Okay, so now we've picked all the different read lengths so we can get the maximum number of DNA binding proteins. You could have gone through this yourself and just seen how many samples there are at each different type, and we can see at 100 there's a lot more, 261. Okay, so we have 566 Chip-seq experiments, which could be duplicates. It also has 34 controls and 15 things. Usually each of these will have at least 2 replicates based on ENCODE standards. Okay, so I think that's all we need to look at for this class. And we'll get into these other details, but this is just a way that you can now browse this data really quick, pick the things of interest, the cell type of interest. And now we can click on, I need to zoom out to see, there's 2 things you want to look at here, is the report. So what ENCODE has done is they have an API, or advanced programming interface, that is saying, okay, if you're interested in this data, I'm going to show you where all the file paths are so you can download them. And we're going to do that in the 3rd part of this class today. And if you want to download them, here's the properties you're going to want and such things like that. So if we look at this and we want to download a report of what this data would entail, you can see a bunch of information here. And that's really great. So they're telling you the things you kind of want to know, especially things like replicates. So if there's one sample here, it's telling you that it's in this cell type HepG2, which we picked. That's good. What lab did it, what the age of the sample was, and what the replicate ID is. And we'll get into more of that as we go towards a design file. So we're really in the basic first steps of designing an experiment. We're saying, give us all the data that meets these requirements. So this is great. It tells us the files, and this is a bunch of metadata, if you will. And that's really what an API does, is it says, here's all the things you might want. So now let's look at the columns here. Okay, it's got everything we wanted. We want, what the ID is, replicates, and making sure that controls is clicked is important. We also requested that MD5 sum, which we'll get into in the next lesson, that is there to make sure when you download the file, you get the right file. There's a way of checking that, actually, and we're going to do that today in part of this lecture in the next video. So we want the controls just so we have, when they give us all the information about the assay we're really excited about, we also want to know what its controls were. And where those files are located, so we can go get those when we make a design to say, here's the control and here's the experiment. Remember that input file we looked at in the last video, where the DNA binding peaks were above it. We want to know where those controls are for which experiment. And then ENCODE has a great way of connecting to make sure that the sample or DNA binding protein you're interested in, where its exact control is and how to link to that very easily. So, okay, so we want to view these selected columns. Okay, great. We're going to get back to something that looks very similar. We're just going to have an extra column now down here, which is the controls. So this will give us the actual file location and name, which we'll get into more. Don't worry if this doesn't make sense right now. It's just important to understand that it's very well documented, as you can see. There's a lot of information here. what the idea of the experiment is, what the name of the file is, what the files are, where their controls are and their replicates. And this will all become really important later in the course when we use Bash and R to take this information and turn it into something we can send through a pipeline and analyze all of this data. It might take 6 days, but it'll work. So that's what we're working towards. And so it's okay if it's a little confusing right now. It's just that we're tracking all of our data. So let's download this and see what we get. Okay, cool. So we have a new file here, and it's downloaded directly to our computer. And we're going to look at this in Excel. We're going to try not to use Excel as much as possible for the purposes of this class. But really what we get is a nice tab delimited file. That's what the TSV is there. And we're going to use this file actually to do some Bash on it and learn some of the basic commands in Bash, as well as using this kind of stuff in R to how do you understand a file from a computer's perspective. But for now, looking at Excel is absolutely fine. We can see that we have the experiment, we have its ID. This is all very small, I realize. So we can see the experiments, what its name is. Really, it's the same as the accession number here. And then we'll get all the way back to one of the more important columns is where are the controls? Where is the data that controlled this experiment? Okay, so great. We're going to need that in our first R Markdown lesson in the next video, where we'll go practice seeing what it's like to download some of this data and how do you check to make sure you got the right data. Okay, enough of that. And so one other thing we need, and the difference in what's missing here is not where the files are. And so we need to know where ENCODE has made a very nice URL system where they'll give you a web link that you can go, and if you click it, you'll download the file. And we're going to show how a computer can do that multiple times and check to make sure that it got the right file before it finishes. Okay, so this sort of ends the idea of the report. We've downloaded the file, and now we need to go to get where the files are and their URLs so that we can use the computer to go download a bunch of these files. So what we need to do here is after download TSV, we go to list. So we're listing all the experiments. And now we can hit this download button. Don't be scared. You're not going to download all of this. This is 2.1 terabytes of data right now. You're not going to download it. But what you are going to be instructed to do, let me make sure I get that again. So you go from report, you click list when you're in the report menu to download. We're going to go over this in our R Markdown file of what this is. And it's really nice that ENCODE just provides you with this line that you can put into any terminal with the file it's going to download right now. And you'll get all this data put on your computer. Make sure you have enough room. It can get big when you do things like this, but we'll go through that step by step in the next video. So let's click download. That's great. So now let's see what we got. We got a thing called files.txt. What is that? Okay, great. And we're going to use this in the next video, and we're just about ready to transition there. But what we can see is those file names, these weird looking file names like ENCF, but we see this normal web looking address shown here that we could just put in. If you put this into your browser, it's going to download it. So don't do that just yet. But this is just a URL that says, here you go ahead and act on download to download this file. And it's saying this is the file it's looking for. So if we just simply copy and paste that and open up our TSV report that we just got in Excel, we can see here, if we make this bigger, we'll put in that file name and there it is. Okay, so we can see that that file actually exists. It's one of our experiments, and it's one of the replicates. We'll get into this more later, don't worry about it, but it's good to know it's there. So we can shut this, and basically this is going to allow us to download all of our files. I think there's 1,099 here. So that's kind of cool. If we with one command in the next video, we could download download this entire list. We're not going to do that. We're going to practice with just 2. And more importantly, what do you do once you download it to make sure you got the right file? What if the internet glitched and a little piece was missing? We'll go over that. So what we've seen from this lecture or this part of the video, that we can go to the ENCODE portal, type in the type of data we're interested. We could have clicked rna-seq at the beginning, or whatever type of data you're interested in, click through it, and you'll get 2 files that are important. One is the sort of metadata file, or how the API is saying, I know where all this data is and I'll go find it if you tell me what you're looking for. And that's what this nice clicking menu was. We got to just simply click through a bunch of information and pick the data we wanted, print it out, and in the list view, we can download to get the URLs, and in the report view, we get all the metadata we need with this, except for one thing, which is the MD5 sum, and we'll talk about that in the next video. And that's a great way to transition from now we have this data, let's go download it. How do we do that? And what should we think about? And we're gonna start to use the terminal for the first time. We're not gonna move into our Fiji, our Fiji server yet. So we're gonna do this just from your terminal on your computer. In the next video, check it out. It's also on the GitHub, the 01 encode.rmd, which is also in this lecture. So if you pull up that file, we'll walk through it on the terminal and download data from ENCODE and see what it could be like with a couple examples, how you can reiterate this and get thousands of samples all at once. All right, see you soon.